Thank you very much for the introduction, and hi everyone. So this talk will be on zooarchaeology. We will look at animal remains from some case studies, trying to reconstruct and interpret cultural and environmental variables which may have affected animal food production at these sites, relying as well on comparative evidence from other regions of the Mediterranean. This is a brief outline of the talk. As some of you may not be familiar with zooarchaeology, I will briefly introduce you to the discipline and the main analysis that we run. We will then move to the case studies, Caral Hamra in the Fayyūm, and we will look at two main issues: pigs, mainly their absence at the site, and fish and fish products. And then we will try to understand the potential implications of these two, of the results of these observations on regional food economy. So zooarchaeology is a branch of archaeology which looks at animal remains from archaeological sites. So it tries to reconstruct the so-called human-animal relationships, which is very much a multifaceted concept. Animal remains can be literally a mine of information. They can be used to tackle several different research questions on animal husbandry, which is what also we will look at, domestication, the use of natural wild resources, social relations and status, manufacturing activities, diet, of course, which we will be looking at, and so on. So the material we look at most of the times is animal bones and teeth, of course, from mammal species, bones from fish and bird species, mollusks sometimes. In exceptional circumstances, we may have soft tissue preservation as well, but this is not what we will be looking at. Most of the times it's basically bones and teeth. And what we look at is actually a minimal fraction of what was once there and of the animals which interacted with humans at given space and time. And so all the analysis we carry out must take into considerations a range of biases, and we are constantly struggling to develop recovery strategies and recording methods adapted to each case study, which can better be used to analyze the sample. So I wanted to briefly go through some of the basic analysis that we run, also because some of these analyses will then be used in the case studies. Obviously, the first one would be the frequency of taxa, or relative importance of different species is something we almost always want to look at. And not just for knowing what was there in a specific place and time, but also looking at trends, intersite or chronological or spatial, or a combination of these. So here, for example, you have a site from Iron Age and Roman Britain, and you have the frequencies of the four main domestic species. And you can see the usual trend of cattle frequency increase through time during the Roman period, which is something we see pretty much everywhere at Roman sites in Britain and the northwestern regions, provinces more generally. Aging is also a very important source of information for us. So, for example, we look at the tooth eruption stages and wear stages. In this case, you have a sheep mandible up here. And what we do is looking at all mandibles from a site, putting them together to reconstruct what we call kill-off patterns. So a graph like this one with age categories and frequency for each age category, which shows how the animals were used. And this is important because if for a given species, for example, sheep in this case, animals were being reared for a specific product, so milk or meat, the animals would have needed to be culled at specific age ranges. So here you have 3 examples from a site in Turkey, Ashvan Kale. It's the study based on which the method I'm using for recording aging for sheep is based, by Sebastian Payne. In this case, you can see a sudden drop in the animals which survived at very early stages of their life. These represent lambs being culled, mainly male lambs being culled very soon to maximize milk exploitation at the site. In these other cases, you can see that the drop is more gradual, but basically it concerns the subadult stage when the animal would have reached its optimum weight, so it didn't make any sense to keep it for any longer if your aim was that of producing meat, and so the animals are culled, and then very few of them survive in later age stages. In this other case, you can see most of the animals surviving into elderly ages, and these are sheep, and so in this case, this is a typical pattern of sheep herding for focusing on wool production. Now, these are sort of very specialized patterns. Most of the times, obviously, what we get are mixed patterns. So, because obviously, in several human communities in the past, it would have made sense to have a generalized exploitation of animals. Carcass processing is also important, and there are several analyses that can be looked at to investigate carcass processing. On Tuesday, I used the body part representation with fish, looking at crania and vertebrae. Here you have an example with cattle, with the elements grouped in with head, upper forelimb, lower forelimb, and the same for the hind limb. And you can see here, for example, that there is a high incidence of cranial elements and lower limb elements, and these would represent the waste from primary butchery, typically, where the bulkier, less meaty parts would have been removed from the rest of the carcass, so the initial dismemberment phase. This is exactly the opposite, so sometimes we have a major frequency of meat-bearing elements, which is telling us that it is actually secondary butchery waste, so the further dismemberment and portioning of the carcass, and so on. Again, like with aging, several times we do get possibly mixed patterns, where which would tell us that the animals were processed whole and from all stages at the same place and disposed of in the same place. The butchery marks are also often left on the bones while when the animals was being processed, and they can also tell us a lot about the technology employed in carcass processing. Based on intensity and standardization, we can see whether the pattern was like an intensive, specialized, large-scale process of butchery, or more an ad hoc butchery of animals, and so on. For the same reason, burning marks can also be useful to reconstruct carcass processing and other taphonomic processes. And also waste disposal practices can be investigated, depending on where, for example, we find the animal remains on the surface nearby dwellings, or in ditches, or in pits. And we can also look at the frequency of gnawing marks produced by carnivores, typically dogs, which were around the site. If there are many, obviously, it means that the material, this food waste, was going around lying on the surface for a longer time, while if we don't have any, we can think that, and we know there were dogs, we can think that there was actually a systematic disposal of the remains in specific pits or ditches. And then biometry, finally, which I'm very fond of, and I very much like, and basically what we do is plotting absolute measurements in, for example, scatter plots or histograms, or look at them statistically. And we look at clusters, outliers, range of values, and so on, and they can tell us And biometry can tell us a lot. It can be used to tackle several different research issues, from the improvements of domesticates like the one we see in the Roman Empire with cattle, but also with other animals. Reversal processes of size decrease, again, something we see in the early Middle Ages in Europe, in some regions, different herd compositions, so this being related to the sexual dimorphism of several mammal species. We can see the presence of distinct breeds, types, or subspecies, investigate wild resource targeting, and so on. And these two research question is something that will come up later in the case study. And just a very brief mention of these analysis, which became widespread in the last few decades. Isotopic analysis, the proportions of some isotopes in the bones and teeth of animals can vary based on variables such as animal feeding, climate, geology, and so on. So they can be used to tackle all these research questions. And then, of course, genetic analysis have been used to reconstruct domestication of mammals and to track mobility of animals and so on. So after this brief introduction to what we do, the main analysis that we can go through during the process of studying an animal assemblage, let's move to the case study and region, which is that of Qara al-Hamra. Here you have a view of the site, sort of. And what we will do is looking and trying to understand the cultural and environmental variables which may have influenced the faunal assemblage at the site, and this will allow us to tackle some of the issues on animal use in the classical Mediterranean. We will look at different methods and propose preliminary interpretations. So Qara al-Hamra is located here in the north of Lake Karun, in north of the depression of the Fayyūm in northern Egypt. In these regions, since Ptolemaic times, works of water management allowed to reclaim some of the land, and the region became densely settled with towns and secondary settlements like the case study of Qara al-Hamra, which is located in a peripheral area north of Lake Karun. So it would have been less fertile and more arid than the rest of the depression. The aims of the Northeast Fayyūm Lakeshore project were those of responding to modern threats to archaeological remains, mainly from agricultural activities, but the main aim was that of trying to understand the role of secondary settlement within the network of larger towns in the Fayyūm. So to see how the settlement developed, agricultural practices and animal exploitation at the site, and so on. This is a magnetic survey of the site with interpretation of the layout of the site. And these are the trenches which were excavated. Trench 1 and Trench 2 were part of probably workshop areas, while trenches 3 and 4 revealed some street sections with probably private buildings. Both local and imported ceramics were found at the site, imported from other regions of Egypt and of the Mediterranean. And these ceramics and coins allowed to date the site to the 1st century BC to 2nd, 3rd century AD, so late Ptolemaic, early Roman period. There is a lot of ongoing research at the site. There are field surveys recently carried out. The 2018 finds processing season focused on pottery, coins, archaeobotanical remains, anthropological remains as well, and zooarchaeological assemblages. And publication on pottery is by Anna Ringheim, was published, and soon will be published the one on the archaeobotanical and zooarchaeological remains. So now moving on to the zooarchaeological evidence, the majority of the remains from this site were fish, as you probably remember from Tuesday. There were also quite some mollusks, but naturally occurring in the sediments mainly. Some wild anatids, and as you can see, there are very, very few mammal remains, just 160, part of which only could be identified. And this absence, or not absence, but dearth of mammal can be a useful starting point to try and understand what was going on at the site. So the reason why the mammals are missing could be, for example, just related to excavation areas. So perhaps in those areas there were not facilities for disposing of mammal remains, so there may be a bias related to excavation choices. It may be related to waste disposal practices, so something similar, perhaps the bulkier waste from mammals being disposed of elsewhere outside of the settlement, so we didn't find them. Or that there could actually be a genuine focus on other animal resources, so it could be actually a real pattern, and so the site would have been mainly a fishing site. If we look at the mammals, there were some caprines, cattle, some equids, the skeleton of a dog, and so on, but none of the 160 mammal remains was compatible with belonging to pig. And pig absence in archaeology is quite a hot topic, let's say. And we will now try to see how we can try and interpret this absence. And this will take us through considering both environmental and cultural options which may have caused this absence. Always bearing in mind that obviously this relies on what we could achieve during this research season, and that we don't know what perhaps may be found in the future. So considering the peculiar, less fertile environmental conditions on the area north of Lake Karun, I thought to start from the basics, so from the environmental requirements of wild boar, which is the wild progenitor of domestic pigs. So trying to understand if the environmental explanation can be a way to explain the absence of pig at Kara Alhambra. Wild boars are omnivorous animals, so they have a flexible diet. As you can see, they have quite a wide distribution in Eurasia and in part of North Africa as well. So they have quite flexible habitat requirements. However, they have limited sweating functions. And so with temperatures above 30-35 degrees, they need especially tree cover and abundant water and mud to survive. And so they tend to prefer woodlands in areas of moderate or high rainfall, or in any case, moist microenvironments within semi-desertic environments. These requirements were passed on to the pigs, which also need vegetation cover and water sources, especially with temperatures above 35 degrees, which are often the case in Egypt. And this may suggest that pig herding is less successful in relatively hot, dry environment. So the environmental explanation could apply. And this importance of tree cover for the survival and the thriving of pig populations is very much reflected on the traditional, let's say, pig husbandry practices that developed in temperate regions like Europe, for example. Here, pigs were often kept free range in the woods to feed on the acorns and chestnuts and whatever else they could find, either seasonally or through quite a prolonged period of time during the year. And so this highlights basically the essential role of vegetation covers for pig herding and integrated use of domestic animals and natural resources on part of past communities. And at the same time, it tells us about the importance of approaching this topic multidisciplinarily. Here you have some example, a couple from Italy, the famous piglets from Sardinia and New Forest in the UK. Obviously, these conditions would have, in several regions, not applied to ancient Egypt, but I just wanted to highlight the important relationship between pig husbandry and forest, which is something that today we tend to forget. And because of this, I also wanted to very briefly mention a case study from medieval southern Tuscany to better understand the strict human-pig environment relationship. This is Curtis Regia, so a major administrative and redistribution center from the Middle Ages. And as you can see, it had really very much a strong focus on pig, almost 80%. If we look at biometrical analysis to make sure that it's actually pigs and not wild boars, considering the overall setting of the site and nature of the site, we can see that based on the standard, which is the one of a population of domestic pigs, most of the values from teeth plot to the left, so they are smaller or more or less the same size, so we can be quite confident that we are dealing with a population of domestic pigs. Perhaps these two outliers to the right here, they may represent the only two wild boars in the assemblage, but mainly we are dealing with pigs. And if we look at the aging analysis, so the mandibular wear stages, very often with pig, we have a peak on the subadult age category, which is when the pig reaches its optimum weight, and so it is usually culled. Here, there is quite a generalized pattern with immature subadult and adult pig, and this is what often happens in the case of free-range pig herding, basically because you have much less control on what pig is what and what age it is. Ethnographically, we know from Sardinia, for example, that sometimes they even these domestic pigs have to be hunted, shot down. So it is a bit more difficult to have to be able to select the specific pig at specific age. Alone, this is not proof that the panel, the free-range pig herding was being practiced, but it is a suggestion that it may be happening. And if we look at the complementary evidence from geomorphology and anthracology, we see that the habitat there had abundant water, and so it was very well suited for pig herding. And anthracological analysis suggests that there were several oak forests surrounding sort of the settlement, and so not just favorable for pig herding, but favorable for free-range pig herding. This is just also from microfaunal remains, the fact that we have species that highlight the presence of both forested and open areas and abundance of water. So all this evidence together may hint towards the fact that free-range pig herding was being practiced at the site. Now, however, moving southeast, quite very much southeast in the Fertile Crescent, here you have a case study from the 4th millennium BC, and all this relationship with between pig herding and vegetation cover becomes more evident because environmental conditions would have been more extreme, and so to a point that they would be actually determining whether pig was present or not at specific sites. Here you have in this map assemblages with little or no pig at all in white, and the black ones with a higher frequency of pigs. And you have the dry farming limit, which in this case relies on modern estimates, but in any case it does provide some sort of delimitation. And we can see that most of the white dots, white sites, are to the south, and the sites with pig are all to the north of this line. So clearly something was going on here, and the environmental explanation seems to very much to apply. There are exceptions, and this because even in semi-desertic areas, we can have most tree-covered microenvironments which were used and exploited for favoring some sort of pig herding. Now, with this in mind, moving back to Al-Kharal, we know what was more or less what was the environment north of Lake Karun. In relative terms, it would have been more arid than other parts of the depression. And the potential implication regarding vegetation cover is clear. It may have been much more reduced. And so perhaps the conditions were unfavorable for pig herding. So the environmental explanation may apply. However, we have also to consider the cultural variables. So basically, what if pig was absent because they were not much into pork? And this is obviously something important to consider. And if we look at the faunal assemblage from Dime, which is just to the west of Karal Hamra, again also to the area north of Lake Karun, there too pig is absent. So perhaps this is supporting what we see in Karal Hamra. And papyri also tell us that most of the village-owned land was located south of the lake, perhaps because it would have been more productive. So, however, as soon as we move to Karanis, which is here, more within the more fertile area of the Fayyūm, we see that the assemblages have a lot of fish, but also quite a lot of mammals, and within the mammal remains, pig is basically the dominant species. And so this is a very, very strong difference compared to what we see in Karal Hamra and Dime. And we don't have other assemblages from the Fayyūm so far, but still this contrast, and you see that the sides are even quite close. It's very strong. Karanis was an urban settlement, so there would have been high meat demand, and pig is very good at meeting this demand. It's a very fast producing source of meat and fat. At the same time, Karanis would have been settled with Roman communities and Roman veterans, and so the famous Roman passion for pork could also add a cultural factor to this, explaining this very high incidence of pig in this settlement. For the Fayyūm, we don't have other assemblages, but we have plenty of papyrological evidence referring to pig. And the first thing that we can consider is that all these references refer to locations located south of Lake Karun. So again, possibly a confirmation of what we have concluded before. In Philadelphia, there is mentions of local pig breeding, trading, the pickling and selling of cured pork. We have in two different settlements reference to a pig tax, so it looked like it would have been something important. In Ewem area, we know that piglets were used in lease transactions. there are instructions for pig keeping and pig feeding and so on. In Magdola, pigs at some point were stolen from a tamarisk enclosure. In Philadelphia, and probably elsewhere in the Fayyūm, pregnant sows were kept indoors and even within the house itself, because would have been a precious good. We have also a reference of pigs contributing to threshing the wheat by trampling on the threshing floor. And some authors, although there is no written evidence to support this, some authors suggest that pig could have contributed to the process of land reclaiming from the lake by uprooting and destroying the marshy vegetation as they do when preparing it for cultivation, although this is a suggestion. If we zoom further out in looking at Egypt more in general, we see that since the Neolithic, pork was quite regularly consumed, so it was popular meat with fluctuating fortunes, but still it was there. And we will now focus on the Ptolemaic and Roman periods. And here I've arranged the same assemblages by chronological periods. And the pig average frequency is 32%, so it's quite high. It's impossible to say chronological trends because the majority of sites are from the Roman period, so there isn't much to say, but the distribution is also quite heterogeneous, as you can see. So the same assemblages were rearranged by site typology. And here you can see that military sites, which are Roman military sites, they all plot. Sorry, I forgot to mention how to read this graph in case you're not familiar with it. So each dot is an assemblage. For example, for this dot, you have this frequency of cattle, this frequency of sheep, and this frequency of pig. So you see the frequency for the three species plotted in this triangle. And so, as I was saying, the Roman military sites all have a lot of pig, on average 60%. The rural sites here are dominated by caprines. So it looks like there is obviously the cultural component coming up here in determining the frequency of pigs at these sites. Just a brief mention of Dime, which I was mentioning before, the site next to Kara Hamra. It has a lot of cattle, animals which require a lot of water to thrive, but these animals come from a ritual context in the site. So we know from the papyri that cattle was being sacrificed and its meat redistributed to the population. So it's an area from the temple, I think, and so it's a peculiar case. And again, the same assemblages were reorganized based on the regions. And there is something going on also in this case. For sites in the delta, the pig average is 26%, which is good. But especially they all have quite a high frequency of cattle, or even the ones down here have quite a good frequency of cattle compared to the other ones. And this also may be related to environmental conditions in the delta, grazing and pastures being much more abundant. For sites located in the Marmarica and the Red Sea coast, less fertile, we see a predominance of caprines in most sites. There are some exceptions here, but these sites, these assemblages are from Berenica, including one military phase of Berenica, so then that may explain this plotting down here. And there is a very contrasting trend for the eastern desert, which seems to go against everything we have said so far, which is the fact that pig is present in very high quantities, 74% average. So what was going on here? Obviously this area would have been quite unsuited to any sort of animal husbandry apart from caprines. and certainly for pig herding. And the interpretation is that these were Roman military and quarry sites, which would have been provisioned more or less regularly from both the Red Sea coast and the Nile Valley. And pigs could be brought on the hoof, but most likely and more often as preserved products, so as cured meat, as either whole or portioned. And this issue of the cured product brings us to another important aspect of Karal Hamra of the site, and in general of food provisioning in Egypt, which is that of preserved food products, which played a key role in ancient Egypt as well as elsewhere and in other chronological periods. So how do we detect preserved food in antiquity, which is something we'll look at after this? We look at bone remains, both from production sites and from consumption sites, so expecting to find different evidence in these sites. We can look at processing facilities like these fish sauce production site in Israel. There is plenty of historical evidence as well that give us information of how this was performed. And we know from zooarchaeological studies the key role of food in food provisions to the army in Roman times of these products. So we move back again to Karal Hamra because, and apologies for those who attended the workshop on Tuesday, there may be evidence that cured fish was being produced at the site. So until now we have seen what was not going on and why with pig. Now we see what was happening. The fish assemblage is dominated by catfish, as you can see here, but there is a much clearer predominance of catfish in trench 1 compared to the other trenches. And if we look at cranial and postcranial elements and their distribution, we see that for catfish there is a more clearer prevalence of cranial remains, and this has been observed in different sites, and it is known that cranial elements tend to survive better for catfish, and so this may in part explain this pattern. But if we look at spatial differences within the site, like comparing trench 1 with trench 3, we see that in relative terms, in trench 1 there are many more catfish crania. And within trench 1 there are some contexts which are made up exclusively of crania, including the very abundant samples, like the one which produced this set of partly articulated catfish crania. And then third anomaly regarding trench 1 is biometry. Most of the fish were small and small to medium size. Here you have the distribution of standard lengths at trench 1 and in the other trenches. And here you have an example of the dentary, a jawbone, to understand what was the difference in size, what could have been difference in size in the fish from these two areas. So I think this could be evidence suggesting that cured fish was being produced in Karal Hamra. The trench 1 focused on catfish, which is not alone evidence of production of cured fish. Usually pickled fish was being made with using different fish species, but still it is a suggestion of something specialized going on in this trench. There is a strong prevalence of cranial elements, which again is not alone evidence for the production of pickled fish, but still it's a consistent activity there, and the removal of the head could have facilitated the dehydration process and has been observed elsewhere. But most importantly, there is a clear prevalence of small individuals. This may be the result of seasonal activity, these smaller fish being caught when the smaller fish were more frequent, but it could have been the result of deliberate selection, so the smaller fish being faster and easier to salt dry, or in combination with this, the small-sized fish we know that were preferred in some products like salsamenta, which was a cured fish product where small fish were used, medium-sized fish were used, but in this case they were chopped in pieces, and perhaps it would have made sense to remove the head for this fish. And so this actually may, together, this evidence may suggest that something was going on in terms of production of cured fish in Trench 1. In addition, we have salt availability. Salt was very much available in Canal Alhambra. It was harvested in the Fayyūm. The site itself was probably along the salt trade route from the Western Oasis to the Nile Valley. And we have abundant comparative evidence from Egypt. And most importantly, we know for sure that from biological evidence that cured fish was being produced in the Fayyūm. Some workshops are mentioned, as well as demands for the delivery of pickled fish and so on. So this was certainly happening. The point is to understand whether Trench 1 in Canal Alhambra could have been one of these production sites. And I just wanted to make a brief note on the fish and mollusks from Canal Alhambra, because they might slightly contribute to understand environmental conditions at the site. We only have freshwater fish, catfish, and hersiforms, and we have only freshwater mollusks, gastropod and bivalves, except from the land snail, Eremina here. They are all, or almost all, natural occurrences, so they would have been present in the sediment. They often also ended up in the mud bricks, which then melted away during when the site was flooded. And so this, what this is telling us is that the lake would have been a freshwater lake during the occupation of Canal Alhambra and in the following inundations. And this is important because we know that, very well know that now the lake Karun is very saline, and there has been a process of salinization that occurred through time. And so this can contribute to the several studies which have been made on trying to reconstruct lake levels and lake salinity in the Fayyūm. But the archaeological evidence may provide a sort of a microchronological scale for these reconstructions, which often tend to regard wider time periods. So in conclusion, the evidence we have seen suggests that in Al-Kara Alhambra, the focus of the animal food economy was on fish, with very little contribution from animal husbandry. Perhaps we have to see what will happen in future excavations, and again, perhaps with the absence of pigs, most likely due to environmental conditions and to the fact that pigs could actually be reared just in the southeast of the settlement. So there is a clear difference with nearby Karanis and the Fayyūm in general, and this was the explanation, perhaps. All these activities would imply obviously an adaptation to environmental conditions and the optimization of what was actually available at the site, so fish and salt. And this would have implied integration of micro-regional food economies, which could have been part of and played a role in the wider scale economy of Egypt. So we have just started reconstructing and understanding what were the human-animal environment relationships in the Fayyūm, and to understand the cultural, environmental, and economic variables that would have influenced it. we need an interdisciplinary approach in order to understand each of these variables, to apply comparative analysis on different scales, and obviously it is essential to acquire further evidence. There is ongoing and future work in the area in Al-Qara al-Hamra. Work is being carried out on the site layout, on different categories of archaeological remains. Possibly there will be other excavations. Regarding the material from Karanis, the recently funded Agros project will look at archaeobotanical and zooarchaeological remains on the site, as well as written sources and complementary evidence to reconstruct the nutritional characteristics of the raw products and the prepared foodstuff. And hopefully there will be more work going on in the Fayyūm in general. So thank you very much for listening.
Yeah, thank you very much, Mauro, for this interesting talk. We now have time for some questions. I wanted to first also ask a question. Do you think it's possible that people were just exchanging with Karanis for their pork? Because if you have prepared pork products like salted pork or something, a lot of these recipes are called for that to be put in pots, and then typically only like what we would call, say, the fillet of a pork chop can be salted, for instance, and then you wouldn't find the bones. And would that be part of the solution? That because they're like 5 or 6 kilometers apart, I thought.
I think
Karanis,
it would make very much sense if, as we say, that within a possibly integrated food animal economy, it would make perfect sense also with sausages, which were basically the leftovers of all the process, which could have been traded without leaving any evidence really at the site. So yes, that's what I would find sensible to think, yes.
Okay, yeah, thank you. And then a compound question. Wild taxa, do you find any wild mammals being hunted, say, on the fringes of the desert, or do you find any exotic taxa that should be non-local?
Nothing. It was all local animals, all domestic animals. No wild mammals at all. But the sample size is quite small, which is interesting in itself, but also a problem in terms of understanding sort of the actual spectrum of species present there, and that could have been a target. Yes.
Okay, thank you. Are there questions in the room? Philippa?
What's the sort of, I might have missed it, but what's the kind of social status of the site or the size of it? So I like the idea of it being like a little local adaptation, wondering about the people.
It would have been an agricultural village, yes. There are houses comparable, some houses have been compared to structures in Karanis, if I remember well, but it would have been a productive site. Until recently we thought relatively small sized, and then perhaps who knows what will come out in the future. There have been some field surveys, but there is a lot of ongoing work as well. So.
Are there any other questions? Annette?
Oh, thanks, Mauro. It was really a fantastic talk. I had a question about using pigs for threshing. I thought that was a really interesting source, and I was wondering, does it mention how the pigs would have been used? Were they attached to a threshing sledge, or would they have been around, allowed to wander and trample the ears a bit? And my related question is, well, because pigs, they like to eat, and you don't want them, eating up your grain. Would there be a way to also prevent them from doing that?
Yes, I have to say this is an interpretation based on a brief reference in a papyrus, which mentioned that pigs were put on the threshing floor.
Okay.
And so, how, why,
okay.
It's up to interpretation.
That would be really interesting.
Yeah.
It's the first reference I've heard of it. So thanks for.
I will recover the reference to the specific papyrus.
Thanks.
Okay. There are no further questions, then I would like to thank Mauro again.
Okay. Hi, Claudia. Could you unmute your microphone and share your screen with us, please? Okay. And Christian will begin his introduction now. Thank you.
You all set? Yeah?
Yeah, you see the presentation?
Okay.
You see me?
Yes, yes.
Perfect.
Okay, so it's my pleasure to introduce Claudia. She got a master's degree in agricultural sciences and technologies from the University of Padova. She worked on the resistance to rhizomania in sugar beet. So that's a name I learned recently. In Italian is barbabietola da zucchero. No, zucchero?
Barbabietola da zucchero.
Zucchero. Barbabietola. In 2020, Claudia obtained a PhD degree from Padova University. She worked on the genetic resources from the beta genus, sugar beet. The Italian, the Latin name is Beta vulgaris, and Claudia looked to genetic resources in the whole genus and related SNPs, so single polymorphism protecting against rhizomania. And during that time, Claudia did an internship here in Belgium, in Tienen, in SOS van der Haven. This is a leading global player, a company in the sugar beet seed sector. So she stayed there during 6 months in Tienen. And for 1 year now, so Claudia is a postdoctoral fellow in the crop production and biostimulation laboratory. And she's working on genetics of root morphology of oilseed rape in response to nitrogen. So Claudia published 11 papers, cited around 100 times, and the floor is yours.
Okay.
So when you're ready.
Yes. So thank you, Christian.
Yes.
So yeah, as Christian was saying, I also work at the DDL lab on the root morphology of oilseed rape, Brassica napus, and it responds to nitrogen. So yeah, I prepared this slide, even if Christian said already everything. I did all my studies in Padova, an internship in Belgium, and now since 1 year I work here at ULB. We'll just give you a brief overview about my PhD project. So as Christian was saying, I worked on the genetic resistance to rhizomania. Rhizomania, or the beet necrotic yellow vein virus. It's a virus transmitted by the fungus Polymyxa abete and attacking sugar beet, causing different symptoms. In particular, a reduced sugar yield, which is a big economical damage. And there are no agronomical or chemical strategies to protect the crop against this virus, only the genetic resistance. So this is what I worked on. I worked on 3 different aspects, on the virus and the soil. So I developed a method for extracting nucleic acids from soil, and using this method I extracted nucleic acids from different soils infected with the beet necrotic yellow vein virus to see if there was any kind of positive or negative effect of this bacterial community on the virus or on the plant. The biggest part of my PhD work was about wild genetic resources. So first, I developed a method for the quantification of rhizomania in the roots based on real-time PCR. This is very important because before than this, just ELISA was used, and I think everybody now is quite familiar with these two tests because they are the same that are used for the quantification of COVID. So the fast sticks are based on ELISA, and if we want to go for the very, very informative and efficient test, we go for the PCR test. So with this virus, with rhizomania virus, it's the same, it's a virus. So I developed the quantification assay based on PCR, and using this, I screened a lot of wild material, identifying one accession extremely resistant to the virus. The first one probably after like 20 years, and found the SNPs associated with this resistance. In the end, during those 6 months that I spent at CES Van der Have, I worked on a bioinformatic project aimed to discover as many as possible resistant gene analogs in the sugar beet genomes. So all the genes related to the resistance. So I came here for my postdoc and I switched slightly. So from abiotic stress to abiotic stress, and mostly from molecular biology to bioinformatics. And here I'm working on the response of Brassicanapus to nitrogen. This picture, everybody probably has already seen it because, yeah, in our lab we use it quite a lot to explain, yeah, the aim of our work. So the problem is that nitrogen fertilization is polluting because big, sorry, a big percentage of the fertilizer provided to the crop is lost in the soil through leaching. So this goes to the water sources, polluting them and causing eutrophisation. So we work on the biological capacity of the plant to assimilate nitrogen, so the nitrogen use efficiency, to obtain a crop which has yeah, higher nitrogen use efficiency. So it's more capable of grabbing all the available nitrogen from the soil, preventing the leaching. So the problem with Brassicanapus is that it has a low nitrogen use efficiency. So this problem is very big for this crop. So what has been seen so far is that like a plant with a more branched root system is more capable of uptaking more nitrogen, preventing the leaching. So my work is focused on the optimization of the root morphology to improve the nitrogen uptake efficiency of the crop. So Brassicanapus, it's a very important industrial crop. It's the second most important oilseed crop in the world. It's used for a lot of industrial purposes. So biodiesel, industrial lubricant, animal feed, but also like it's a edible crop. So its oil, it's used in the different product that we eat, and in particular, as a colleague of ours put on our attention, it's the fat part of the speculoos. These Belgian biscuits are actually vegan, so there's no butter, despite the big quantity of butter consumed in Belgium. But yeah, there's oilseed rape oil. Why do we speak about a system biology approach to answer the problem of the root morphology? it's because it allows a very holistic approach, so we can combine different scientific disciplines. So in my work, I just, do you see my arrow? In my work, I just focus on genomics and transcriptomics, but yeah, the limit to how many we want to integrate is just our yeah, imagination, or yeah, even with metagenomic, even here we could find some bacterial community affecting maybe the root morphology or the nitrogen uptake, or yeah, wherever. So something else about this crop, it has a very big genome. This is the triangle of the Brassicaceae. So basically, all the Brassicaceae come from the crossing of three main ones, and in particular, Brassicanapus comes from the spontaneous hybridation of Brassicarapa and Brassica oleracea. So as you can see, it has a pretty big genome. What I'm working on right now, again, there are 3 projects. I'm working on a review to see the work that has been done so far for nitrogen and phosphorus uptake improvement in Brassicanapus, on a transcriptomic project to identify differentially expressed genes in response to different nitrogen concentration, and on a genomic project, in a bulk segregant analysis between contrasting group phenotypes. So regarding this project, I will give a brief introduction about the topic. I made this figure, which is summarizing the main papers, the main literature regarding the improvement of nitrogen and phosphorus uptake in oilseed rape. So up here, in the upper part, you can see the phosphorus-related research, while down here you can see the nitrogen-related research. Each box is a paper, and I highlighted the paper which show QTLs, quantitative trade loci, for phosphorus and nitrogen use efficiency improvement, and those which are not highlighted are just like supporting research, very much related. So when I started working on this, as I was saying, I was working mostly on biotic stresses, and I switched to abiotic stresses. So I was very surprised to see that, as you can see, there's a lot of material on phosphorus, but very few on nitrogen. And I was very surprised, but the answer to this, it's quite under the sun now for me. Phosphorus is a very important nutrient, of course, and it has a very low bioavailability. While nitrogen, of course, it's important, but we can just fertilize the crop, and we are okay with the nitrogen supply for the plant. So the nitrogen use efficiency, the improvement of the nitrogen use efficiency is not really been studied so far. Only recently it has started to be seen as an important environmental problem. So the number of publications in this topic has been growing. So from this paper, we summarized some QTLs. This has been done with Loic, a PhD from our lab. And so I will show you this project, which is the main one I've been working so far. It's a problem. So for those which are not really into the topic, it's a transcriptomic related project. So after the DNA, the DNA is transcribed into RNA, and here we can see the expression of the gene. So different genes can be expressed differently in response to different stimuli. So also here we did this type of work. So first, as I was saying, we screened a lot of lines, 40 lines, for checking the natural variation regarding the root phenotype into two different nitrogen conditions, low and high. And among these, we chose 6, 2 because of their extreme phenotype on one side with very small number and very short lateral roots, 2 for their extreme phenotype on the opposite side, and 2 with the intermediate phenotype to use as control. Also, the biomass of these plants has been checked. So it's important because in the idiotype I showed in the picture at the beginning, we don't want that we have plants with a very big root system sacrificing the upper part, because this would be translated, of course, in a lower oil yield, so it's not convenient. So we want plants which actually produce a lot of roots without sacrificing the upper part. These 6 lines have been chosen because they have all similar length of the primary root, but extreme phenotypes for what regards the lateral root length and the lateral root number. They have all been grown into 3 different, after they've been selected, they have all been grown in hydroponics in 3 different nitrogen conditions, low, high, and normal. And just these 2 have been selected for RNA sequencing. This work was done from a former postdoc, which did all the phenotyping. I took over the data and I analyzed them to arrive to the differential expressed genes. I followed a pretty common pipeline. So I just screened the reads for their quality. I aligned them on the genome, and I use DSeq to find the differential expressed genes. The result from this, it was pretty good. So from the PCA, we see that the 2 varieties with a lot of lateral roots clustered together, the 2 with the few lateral roots also clustered together, while here we have the 2 in the middle. So I checked for differential expressed genes. I checked those which were common in the 3 data sets, unique for one phenotype, unique for the other phenotype. This is a summary of what I obtained. So a lot of upregulated genes. So I will speak about log fold change. It's the degree of expression of one gene. So if let's say in a control condition, a gene is expressed this much, it's upregulated if it has a log fold change of 1, 2, 3, 4, and it's downregulated if it has a log fold change of -1, -2, -3. So it can be more or less expressed. The problem here is that what I got so far is that the choice of the genes to focus on, it's kind of personal. There are no specific protocols. So someone choose to go for the genes which have the highest log fold change because they are highly expressed, maybe specific for, let's say, the good phenotype, but then also those specific for the bad phenotype are interesting. So yeah, choosing genes to focus on and deciding that those genes are important, it's kind of personal. So what I did, because yeah, the whole aim behind this thing is to understand why a crop, why some plants are special, let's say, why they work well, what are the genes that makes them good, so that we can breed them to obtain population of these plants to grow and sell. Yeah, about the choice of these genes, what I have done to understand what's the meaning of them, I checked for the gene ontology. Each gene is classified according to different function, molecular biological process, molecular function, cellular component, and it has been classified in a kind of universal dictionary of Go term, of gene ontology term. There are also databases like KEGG and Reactome, and in particular Plant Reactome, showing what is the pathway in which each gene is involved. So to understand its function in the organism. So I went for all of these, let's say, databases to understand the meaning of my genes. In particular, for the gene ontology enrichment, the problem is that the genome of Brassicanappus, the last version, it's super new. It's not annotated. So I used this tool, InterProScan, which is an amazing tool actually, to annotate my genes and to obtain their identification with a specific gene ontology term. The result of this is this. So here I show all the all the genes which are somehow differentially expressed, so upregulated, downregulated in the three data sets: high lateral roots, average number of lateral roots, low number of lateral roots. And the size of each bubble, here you can see the three different classification of the Go terms according with the colors. The size of the bubble represent the number of differentially expressed genes in that Go term, while the significance of these, so the highest are the most significant, it's calculated basing on the number of Go term with that name in the whole genome. So for example, here we have this transporter activity, which is super significant. This means, now I'm inventing the number, that for example, if we have in the whole Brassicanappus genome 20 Go term with this name, maybe like 18 out of them are in my data set of differentially expressed genes. This means that this process is very significant, is very correlated to the analysis, with the analysis that I'm doing. So with this, I was pretty happy because, as you can see, we have a lot of transporters, and yeah, this means that actually the plant is working to transporting all this nitrogen. So what I did, I went into the details to see what Go terms are specific for the phenotype with many lateral roots. And I took the 10 most significant ones. Here we have the up and down regulated genes for each Go term. Again, you can see we have a lot of transporters and even something related to nitrogen. While on the others, sorry, on the other side, we have those as control common to the three phenotypes. Here we can see that we have a lot of terms related to nitrogen. This means that, yeah, the plants, the three phenotypes are activating kind of the same mechanism to respond to the nitrogen situation. While I also checked on the phenotype with low lateral roots if there was something interesting specific to this phenotype. As you can see, there's nothing related to nitrogen or to ion transport. But I was pretty impressed from the fact that there are a lot of catalytic activities here, which would be nice to investigate. Why doesn't go? Oh, okay. So I also checked KEG, so the Kyoto Encyclopedia for Genes and Genome, to see which pathway are significant. Again, I divided them according with the phenotype. So specific for the high number of lateral roots phenotype, specific for the low number of lateral roots phenotype, and common to the three phenotypes. I did a research in literature, so all these results that I'm showing since now are really new. So I didn't really have the time to go deep into their meaning. But still, I thought they were quite interesting. For example, here we have a lot of pathway related to the metabolism of fatty acid, and I did a research in literature, and I saw that actually long chain fatty acids are signal molecule activating the ethylene biosynthesis. Ethylene is a hormone known for promoting root elongation. On the other side, on the opposite phenotype, I checked what could be the meaning of galactose. And I saw that it's toxic for certain plants, and it's inhibiting the growth roots, even when used as medium. Again, I checked on Reactome. This is another database for pathways. Here also we have metabolism of fatty acids, so it agrees with the other analysis. And so I also saw that there are kind of two different opposite processes here, but I was not able to find their biological meaning in literature so far. But as you can see, there are a lot of processes involving hormones known to be involved in the plant growth. On the other side, I saw that the phenotype with low lateral roots presents some pathway for the abscisic acid, which is like the stress hormone. So apparently this phenotype is really feeling like the stress of nitrogen deficiency. And I also developed co-expression network for both the two main phenotypes. So these are just the genes specific for their high lateral roots phenotype, and they've been classified. I used Cytoscape, and they've been classified according to their function. As you can see, again, here we have a lot of transports. And on the other side, those specific for the low lateral root phenotype. I'm not going into the details of this because, yeah, it's still very new, so I'm still trying to figure what's the meaning of this. But the idea is, in the end, to have I have some candidates. I would like to add some to this list to validate by Q-real-time PCR. So here we have a lot of transporters, some related to nitrate, and so the idea is to validate to grow more plants. I'm actually growing more plants to redo the experiment and to validate these genes. And what I would like to do is, from the pathway analysis, to choose some compounds to validate as well, to quantify and to see if there's actually a correlation with the gene expression and the nitrogen condition. So at the beginning, I was saying that the plants have been grown just in high and low nitrogen condition. In the end, they have been also grown in normal, but they didn't went for RNA-seq. I would also add this to the final validation process to use it as a control. In the end, I was mentioning also a project related to a bulk segregant analysis. This I have just started it, but I will show what I have done so far. So a bulk segregant, sorry, a bulk segregant analysis. Basically, what we do in this analysis is we have two contrasting parents with two contrasting phenotypes. We create F2 population, and I mean, this population is supposed to have a kind of normal distribution, and we just take the individuals with the two extremes phenotypes, so the two tails of the curve, and we bulk them. So we pull the DNA of all these individuals, and the aim behind this is to find SNP markers, so single nucleotide polymorphism, correlated to the phenotypes. In particular, to find QTLs, so zones in the genome very rich of significant SNPs, so zones which are highly correlated to the phenotype. To do this, I used this software, QTL-seq, which is basically ranking the SNPs. This is a basic representation of a chromosome, and each dot is a SNP. Is ranking the SNPs according to this SNP index. The significance of this SNP index is related to the frequency of this allele in the genome. So an allele which is very different from the reference is super significant, while not too much frequent is less significant. So we map the two bulks, low and high, and it depends on the phenotype. Here I am also working with truths on the two reference parents, and then the software is calculating the difference between these values, so that we have a unique analysis for the two bulks. In my case, I used parent with low lateral roots, a parent with high number of lateral roots, and the two bulks deriving from their crossing. These are the results of the phenotyping. As you can see, we have a good normal distribution for what regards the lateral roots number and the lateral roots length. So we took the two bulks, so the two tails of among the progeny, and we sequenced them. So we sequenced basically 4 samples. Here there's a report of the number of SNPs and indels found in the genome using the two parents as a reference. So some are specific for one parent, some are specific for the other, some are common. The reason behind this unbalance, so there are way more SNPs for Trebisca than for Cadi, is that Cadi is way more similar to the reference genome. But in the end, most of these SNP will end up as with a low significance. So we have kind of the same number for the two parents. These are the results of my analysis. As I was showing at the beginning, we are supposed to have a peak higher or lower than 0.5, which I don't have. So the analysis was not super successful because we have a lot of heterozygotes in the progeny. But what I got is that to find the area of interest, we have to look at the red line, which represents the delta SNP index, and to check when it has a opposite behavior in the two, using the two different mapping parents. So in this case, for example, I highlighted something which could be classified as an area of interest because it's going down here and it has a opposite behavior here. While, for example, this red line here, it has more or less the same height, but it has not the same opposite behavior on the other side. So this is not very interesting. So this is how a good analysis is supposed to be. But still, I was able to find something interesting. So there are a lot of SNPs where the behavior is kind of similar to the two parents, even if they are mostly heterozygote. So this is one parent, this is the opposite parent, and you can see that the parent with low lateral roots, bulk with low lateral roots, bulk with high lateral roots, and the parent with high number of lateral roots. You can see that, for example, the SNPs are kind of correlated to the with the phenotype. In particular, this is gene coding for a kinesin, which is actually involved in the root shape. And what was interesting to me is that there are these SNPs are all in the 3' UTR extreme, which is a target for microRNA. So SNPs in this area can actually influence the expression of this gene. Yeah, the studies like this have already been done in rapeseed. Yeah. Yeah, there's not much to say on that project, but in conclusion, what I wanted to say is that, yeah, there are more concerning about the environmental impact of agriculture. So studies about the impact of fertilizer the number of these studies is growing, and but these problems cannot be faced using just a single approach. That's why we talk about system biology. We can go for genomics, transcriptomics, proteomics, and how many omics we want to add. And yeah, there are actually tools and a lot of databases available for approaching this kind of research in this kind of holistic way. And done. Thank you.
Thank you. Are there questions for Claudia?
I have some, but I'd rather.
If it was too technical, you can ask to re-explain things if you are not from the domain. Yes.
I'll give a stab at it. No, thanks, Claudia, so much. Really interesting talk with, you know, I dabble a little bit with rhizomes, but I think root morphology is a very interesting topic, so thanks very much.
Thank you.
I have a short question and then a slightly longer question. Short question with the naming convention of your plants. Is there a convention you follow? I saw one called Katana and something neuf or jet neuf, but I'm wondering if is there a convention followed here for the naming of your...
No, no. This is true for all the crop. I think it comes from the seed companies. It's a kind of signature of each seed company. So, for example, yeah, in case of Sesfanderave, they name all the accession with the name of animals. And KWS, I think they use all women name, right? Or something
there should be Claudia. I was working during PhD with
exactly.
Then my next question has to do with your plant katana, actually. It had some very attractive lateral roots there. And I was just wondering if you wouldn't mind explaining for me, what type of challenges do you expect? Now they're grown in hydroponics, of course, but what type of challenges do you expect to face in terms of nitrogen use efficiency, phosphorus use efficiency, once you get the plant from the hydroponics to the field context in soil? So what kind of challenges do you expect in terms of its performance under new conditions?
Okay, so I mean many. So actually my plants are trying to grow as healthy as possible, but yeah, it's not really easy to grow plants in artificial conditions. There can be pathogen and whatever. So this can actually be a problem. And then for what regards to the validation of the genes. There's a very big genome, so it's very hard to design primers specific for one gene because there are many copies of the same gene in the genome. And yeah, actually, these are the main problems. Then once you have identified a gene or a characteristic related to that phenotype, it's as a researcher, we are kind of done. It's more up to the seed company. They cross and cross until they have a population that they can sell with that specific characteristic.
All right, thanks very much. We do have one question in the chat from Marco. So
maybe I'm able to read.
Yeah, you can read it from the chat.
How do you want to confirm the role played by the candidate genes you identify to work with loss of function? Yeah, I still don't know. Because I'm having some problem with the growth of the plants. So the validation will be a bit tricky. We'll see. Yeah, right now while the plants are growing, I'm trying to do as much as possible in vitro. So to find some maybe common candidates between all the analysis that I have done, because yeah, I don't know if I will be able then to maybe do it in vivo. But yeah, also working with Arabidopsis would be interesting. Is typing again?
So if it was not clear for the animal people,
Thanks. Okay.
oilseed rape is a cross between a cabbage and a turnip. And the beauty of the plants is that you can cross parents with a different number of chromosomes. Maybe Claudia showed that at some point, a diagram with a triangle. Yeah. So the top is Brassica nigra, it is mustard, and then there is Brassica rapa, it is turnip and Chinese cabbage, and Brassica leracea, all the cabbages like cauliflower, green cabbage, Brussels sprouts. And the beauty of these plants is that they can cross, even though they have a different number of chromosomes, and a cross between a cabbage and turnip gives oilseed rape. And it's super large genome, super complex, because it takes double information from both sides. So the chromosomes, they never pair when there is a meiosis, so they stay all together. Thank you. And Ramji did a presentation a couple of weeks ago on Arabidopsis thaliana. This is the model species in plant biology. So when Arabidopsis and Brassica separated, and if I represent one time the genome like this, when they separated from Brassica, so these are all the cultivated Brassica species, so there was a triplication event, so they got 3 times the information already present in one of the model species. And then you see this in the triangle. We have Brassica nigra here, Brassica rapa, and Brassica leracea. This is called the U diagram by the name of a scientist from Korea. So it's called U diagram. And Brassica nigrus is a cross between the two here, but is keeping all the genetic material from this parent, already 3 times the information from Arabidopsis. Okay, so in total you have 6 times the information. So for one gene you would have in the wild species here, Arabidopsis, you will find 6 copies in the cultivated plants. So that's why it is super difficult to work on these plants, because you have too many information in the genome. And if you just want to study this one, you have to silence this, and this, and this. So that's why we continue to work with that model species, which is just a weed without any agricultural interest, but we can validate quickly the findings in the crops. Okay. So for example, Claudia was showing that this gene, for example, would give a lot of lateral roots, and in order to prove this, we just go back here to this. Otherwise, we have to silence 5 other copies. Okay? That was just for the animal people to explain a concept. And the name of the progeny is called amphidiploids. So number of chromosomes from the 2 parents are different, but they take it as a set without the meiosis. Okay. Other questions for?
Just a comment on that.
Yes.
That's archaeobotanically, that's what makes this crop really a tricky one for us too, because we're often, what we do find left over mainly are seed finds. So, you know, based on that, we can't always tell what crop we're looking at. And maybe if we get an assemblage from a waterlogged context, then maybe we get other plant parts like leaves and things that we can maybe relate to a specific crop, but it's very difficult archaeologically for us to tell what crop. Sometimes with the Brassica rapa, for instance, there's also quite a lot of variety just there as well.
Yeah.
So which one is kind of the big question for us? Just a comment.
And it's quite recent also as a crop. 3,000? Maybe someone else? 3,000 years. And there is no wild species. But you know better, probably.
I know that one of the Ethiopians from 3 or 4 thousand years ago, but that's, I don't know in the Middle East how early the backwards are and the circles from continents as well. I'm not sure.
So there are the other amphidiploids. So if you cross the mustard with the turnip, if you cross the mustard with the cabbage, it will give another amphidiploid. So it's Johnsia and Carinata. And these are used in India, for example, to produce oil.
Testing wild mustard seed deposits at Fayyūm in central Anatolia that's about 10,000 years old. I can't remember if they decided what species that was, but I'm pretty sure they said it was like a wild mustard. But they could see it was well exploited for food because it was a huge deposit in a storage bin that preserved in a charred building. So that might, that's still a wild collective thing, but it's some relationships and also consequences. And know Mark Arts was saying about a wild relative back the last couple of years, I think being in the UK and the Western Mediterranean. This was the first couple of the domestication being in the Eastern Mediterranean, and perhaps the wild relative has a really wide distribution, but it's such a common weed, invasive weed. It's like the relic populations of the wild, just now quite far away from the early cultivation zones.
And the diversity is quite, genetic diversity in Apis is quite narrow. Other questions for Claudia? No more? All is clear? So thank you for your effort. And we wish you a speedy recovery.
Yes, sorry for not being there. See you next week, I guess.
Okay. Bye-bye.
Bye.
With this, maybe there are some announcements.
Oh, yeah. Just a couple. Can everyone hear me okay? So next Tuesday, we have a BrIAS talk by Mauro Rizzetto, 11 to 12, and that is in the campus plane building BC.
6th floor.
Next week on Wednesday, we have another nice workshop, afternoon workshop, on the long-term influence of imperialism, colonialism, and acculturation in agricultural and dietary change in Africa, from 1 to 5:30. And then Thursday is our final event, so our final lunch, and then a set of talks by some guest speakers, hopefully from the Food and Agricultural Organization in Brussels, and followed by a reception at the Bar Pilar and the VUB campus. So we invite you to join us for our events and join us at the reception. So thank you. Those are the 3 for next week.
Okay, so we will close here. Have a good afternoon. Bye-bye.
Bye.